Okay, good afternoon, everyone. Thanks for joining this webinar. This webinar is organized by GINAMO, which is a Biodiversa project funded to study genetic indicators and understand how they can be used in practice. And we're organizing this webinar after a number of stakeholder workshops we have had over the last year with the different countries. And after receiving input from these workshops, we compiled a set of questions that are very generally discussing the use of the genetic indicators, especially in the light of the upcoming reporting for the Convention on Biological Diversity. Let me first introduce our panel today, or they will introduce themselves. Julia, can you please say some words?
Sure. Hi, I'm Julia. I am currently based at the University of Freiburg in southern Germany, and I am a conservation geneticist by training. I have been investigating adaptive variation and the link between environment and genetic diversity in a bunch of different species. And I am part of GINAMO since last year, June, and I'm very excited to be on the panel today.
Thank you. Then Joachim.
Hello, I'm Joachim Mergeay from the Research Institute for Nature and Forest in Belgium. And in this capacity, I'm working on genetic diversity, genetic indicators for policy relevant species. And we advise Flemish government on all kind of aspects related to genetic diversity, especially with regards to Habitat Directive and the Birds Directive.
Thanks, Joachim. And then Sean.
Hello, everyone. I'm Sean Hoban. I'm a senior scientist in tree conservation biology at the Morton Arboretum, which is a botanic garden in Chicago. And I've been working on the genetic indicators since 2020. I was part of the team that helped present the indicators at COP15, where they were ultimately accepted by the CBD. I also help lead a working group in the GeoBon organization, and I lead the Coalition for Conservation Genetics.
Okay, thank you. Before we start with the questions, we've prepared a few slides to ensure that everyone is on the same page regarding genetic indicators. If we talk about genetic indicators, we basically talk about two different genetic indicators. And the first genetic indicator is also called the NE500 indicator within the CBD framework, called the headline indicator A.4. And this indicator tries to quantify within population genetic diversity. And the way it's calculated is very straightforward. You have a number of populations within your reporting territory, and a certain number of populations will have effective population size larger than 500. In this example, there's 2 such populations, and then there's 5 populations in total. So the NE500 indicator is 0.4. And then there's another indicator, indicator number 2, also known as the populations maintained indicator. This is a complementary indicator, and it quantifies between population genetic diversity. And how is this one calculated? Here in the example, we saw that there were initially 8 populations, but there's only 5 extant populations, and while 3 populations went extinct. So the populations maintained indicator is 5/8, or is 0.625. And then we've been working with a number of countries to implement the assessment of these genetic indicators and work towards the CBD report, of which the first reporting is due in 2026. I'm sure many of you are involved one way or the other in this reporting. And what you see here is basically a pipeline or workflow, a general pipeline or workflow for countries reporting on those indicators. The first step in general would be a species selection process where countries decide what species they want to report on, like birds or trees or fungi or butterflies. After that, data are gathered and stored. And the next step would be for each species to come up with a way to delineate populations to determine how many populations there really are or have been. and then the indicator calculation work can start as shown on the previous slide. So that forms the basis of the CBD report. I know that this workflow is extremely simplified and that there's a lot of issues regarding how to do this exactly for any given species. And that's exactly why we want to have this webinar to discuss some points. And we will do this in 2 parts. For the first part of the webinar, we have prepared some questions based on basically our experience within GINAMO. And the first question is whether, well, countries have a species list, and this list might be biased towards some groups where we have a lot of data, because there are national conservation priorities. For instance, a country might report a lot on butterflies. And to what extent is this a problem? And this question is for Joachim.
So yeah, thank you for this question. So I would say it depends on what you want to use the data for. I mean, if you are biased towards butterflies because you have a lot of data on butterflies, it will be most informative on butterflies as the species group. And your indicator values that you calculate as an average across all the species you assessed, they might not represent all the taxonomic groups, but this is something that occurs in all kinds of biodiversity data. For example, in red lists, they are rarely entirely comprehensive as well. So we need to acknowledge these biases, but it doesn't mean that our indicator values become useless. So we're not trying to, I mean, we know it's going to be imperfect anyway. So for each assessment, however, this will tell you something about the status of that species and its population. So already there, this can provide very useful information to policy and management with regards to that species. For the CBD, we're trying to get 100 species and then report the aggregated data over these 100 species, and also per taxonomic group, an aggregation. We need to know what are the limits of these examples. So I could quickly show you an example, but for Belgium, we have data on only 7 fish species for the moment, which means the result for the fish species will be largely dependent on which species were included here. Because so we need to be aware of biases and acknowledge the limitations thereof. But it's better to have good data on a number of species than saying, well, we're not going to show anything because we're biased.
Okay, thank you, Joachim. A follow-up question. Are the indicators even meaningful if there are these biases? How can they be comparable between countries? Question for Julia.
Yeah, that also goes a bit in line with Joachim. So I think to answer this question, we also have to reflect a bit what these indicators are sort of used for and intended to be used for, and what are the benefits of calculating these, right? Reporting on the genetic indicators helps us to track change over time or maybe direct conservation actions. And as Joachim said, is that there's always a chance of bias. I think that is unavoidable because based on maybe existing programs, right? So maybe a country is having a lot of monitoring programs on butterflies or also on data availability. So I think the bias is just there. We just need to be aware of it. And I think comparing between countries is possible, but also it really depends on the species we put on the species list. And I think what I also want to emphasize is that it's also important to sort of to think about within a country, because when we sort of try and track change over time, sort of you calculate the indicator for a certain set of species now, and then you do that again in 29 and hopefully again and again. That means you can really track this change, and that also helps to raise alarm to certain species if they have a low indicator value, for example, and also inform policy and helps them to direct resources or conservation action to certain species or species who need it. So I think it's comparable between countries, but also we should really emphasize what happens within the country. Yeah.
Okay, thanks, Julia. Question number 3. Let's make it a bit more complicated. In some species, we cannot reliably identify individuals. For example, some plants, such as blueberries, spread via rhizomes, and it's impossible to count individuals. We therefore cannot obtain census data, and we do not have genetic data for these species. Can we include them anyway?
Joachim, do you want this one, or should I take this one?
Oh, I'm glad if you take this one.
Okay, thank you. Yeah, I work on plants in my job at the garden, so I'm very familiar with this challenge. I think there's 2 parts to answering this question. The first is about identifying individuals. Yes, you often, for grasses, for some clonally propagating woody plants, you can't identify a given individual, but often we have some knowledge of they don't spread too far, or maybe we have some knowledge, an average plant is maybe 5 square meters or 10 square meters. So you can rather than count the individual, you can count the number of those unit areas that fit in your population and then come up with a very rough population size using that kind of density estimate. And the other important thing to remember for the indicators is you don't need an exact count. So some people come to us and worry about having an exact count. The important thing is a sufficiently reliable count that you can compare to the threshold of any 500. So in many cases, you can get a reasonable count that maybe there's 100 or less likely individuals in this population, or maybe your estimate is there are many thousands, and that is sufficient for reporting the indicator. You just need a rough estimate relative to that threshold. The last thing to say is even if you cannot, maybe there's an instance where you really have no idea of how large an individual is. This is maybe often the case in some fungi. You can still calculate the other genetic indicator that you introduced at the beginning, the population's maintained indicator, and you can report on some things like area of occupation and how that has changed, area of occurrence and how that has changed over time. So I hope that helps. Encourage you to continue to try to work with such species. But then also, if it's really impossible, you can still use the other indicators for reporting on such species.
Okay, thanks, Sean. Next section about population delineation. And the question for Joachim is, how can we define populations in a realistic way when we don't have genetic data?
Yeah, I think that's a $1 million question with regards to this indicator work. There's different ways to do this. So this is an example from a butterfly species. In this case, we did have genetic data, but which corroborated our non-genetic approach. So we have, from species observation platforms, we have individual observations around which we put a 10-kilometer buffer distance, which we consider to be a median dispersal distance for this species of grayling. Well, if you then connect all the overlapping buffers, this helps you to define what are plausible populations at least. And if you have then a big gap in between these two, you can consider them as distinct populations. So in this case, we have two distinct populations. The one in the west at the coast in red is, at least according to population genetic data, smaller than 500. The one in the north is larger than 500. And then you have this yellow dot. This is a singleton, which is probably just an isolated dispersing individual that was in this way found. Now, I have another example for another species of butterfly. It is this Pyrgus malvae, where we have another dispersal distance because we know this species disperses less far. And there you have, especially in the northern part of the country, clearly very distinct isolated subpopulations. In the southeast of the country, you find this one large green blob. Even though, according to our buffer distance, they are some of them which are isolated, we need to take into account that we don't have necessarily a full picture on the complete species occurrence. So here, it seems plausible to consider this more or less as a single metapopulation. Eventually, we will try to use other sources to confirm or to test what we're trying to find out. In some cases, there are clear dispersal barriers in between. For example, in amphibian species, there could be highways or other physical barriers. So in these cases, you can add that information to this kind of dispersal based approach. It's not perfect, but it will tell you in many cases already a lot about the different plausible subpopulation or metapopulations that you have.
So I can for sure like share another example. Joachim gave a nice example about butterflies. So within GINAMO, we are working also on a project where we are trying to calculate the indicators and delimit population boundaries of the bird species listed on the EU Annex 1. And so this is really nice because that's a big data set, publicly available, and but also challenging in itself because, as Joachim emphasized, population delimitation, sometimes it's maybe clearer than other cases. And so in our project, what we're trying to collect different data sources. We're basing this population delimitation on sort of species-specific characteristics with birds. The migratory behavior is very important, so we're also accounting for that, dispersal distances, abundance maps, and in general, like census sizes. And what we're putting in as sort of data or information is if there's maybe subspecies recognized, if there are genetic studies available, and a little spoiler, not for every bird species on the Annex 1 there is genetic data available, so that makes it sometimes maybe a bit tricky to delimit population, but also not impossible. And also we sort of had expert judgments on sort of delimit population. And I just brought two examples and just to show sort of a little bit like how we did that. I brought the white stork. And so the white stork within Europe has a broad distribution, and there's one subspecies recognized, sort of, and it's migratory. And here on the right side, I don't know if you see my cursor, I hope so. On the right side, you see sort of an image about the migratory routes. So there's two migratory routes, which sort of would suggest that there might two different pathways, but based on a genetic study working on different populations from Europe, there was a mixture detected, and so it wasn't really sure if that is now sort of two different populations or one. So that is something we then had to consider as well. And based on this genetic study, because there is a mixture, they also found that the European population is rather panmictic, and there's no spatial structure. And based on all these information, we sort of voted for like one big meta population across Europe for the white stork. Again, there's no perfect way to delimit populations. It is based on information and expert knowledge. And of course, there's not like one way to do it correctly or not. This is sort of how we tackled it. Another example is the Barbary partridge, which is a bit the opposite example. So there's more limited or scattered distribution across some areas in Europe. So on the right side here, you see sort of the map where it occurs. And so there's no recognized European subspecies, which could maybe help us in delimiting populations. And it's a very sedentary bird with very limited dispersal distances. And when we try to find genetic studies to maybe support our population delimitation approach, we only found like a study from Sardinia, but not really range-wide. And that's, of course, also an issue if you only have from a certain area genetic data and not across the entire range you're looking at. And based on that, we delimited 9 populations across Europe because of this like limited dispersal distances and this limited dispersal rate. Because of these little islands, each island was so sort of supposed to be one population.
All right. Then a question for Sean. What should we do if you know a population has recently fragmented into 2 due to habitat loss, or the opposite, when 2 populations have merged due to the removal of some natural barrier? Does this count as 1 or as 2 populations?
Thanks for the question. This is one of the hardest situations for the genetic indicator. I'll first say there's no one answer that will apply everywhere, but I hope that my answer will generally apply in most cases. Fragmentation, let's start with the fragmentation one. Fragmentation into 2 populations or many small populations can cause the appearance of generating new populations. That's the challenge we might be facing. And so this would especially impact the PM indicator because you would have the appearance of many, or at least several new populations when you used to have 1. But the key is, was the historic population lost? So was that entity as it was for fragmentation, has that been lost? That's your reference. So when you maybe have that historic population fragmented into multiple pieces, maybe one of those pieces is lost, but that historic, whole, locally adapted population is not fully lost. So you should be comparing back to the reference historic population. However, some genetic diversity has been lost, so we do need to think about how area of occupancy may have occurred, how area of occupancy may have changed over time. In terms of merging of populations, this also depends. In some cases, that could be a problem, and in some cases not. I think that would have to be dealt with on individual basis. If the merging is such that the local adaptations of those 2 original populations are maintained, so maybe some slight increase in gene flow, but they're still locally adapted, and that local gene pool is still there in each place, then those populations would not be considered lost as such. But if they fully merge, then perhaps some of that local adaptation would be lost. So this is probably the most challenging case. And we don't have a full answer for this kind of among population hybridization potential. So it's a challenging question. I hope the first answer I gave is probably the more common one, and it should apply in most cases. And I guess the last thing I'll say is through both the GINAMO network and other networks, such as the Genoa network, we are activating dozens of experts within countries across Europe and across the world who can be here and available to help. So that's the other thing maybe to take away, is to collaborate, collaborate with those in our network, and we can help interpret particular cases. Thank you.
Thanks, Sean. Then the last section of this first round is about indicator calculation. And I have a question for Sean again. if we're talking about calculation for a single species, in cases where some of the populations have unknown census size, or the range includes 5,000 individuals, how do we best account for that uncertainty when calculating the indicator?
Thank you for whoever submitted this question. It's a good one. We come across several types of uncertainty. The first type is where you maybe have disagreement between data sources or experts. So maybe you have interviewed a person who's an expert on the species and they say population is 200. And then you go to the forest inventory report or some official document and you find a different figure. Maybe this report says it's 2,000. So in this case, there's quite a big difference. If the difference is very small, then you could take an average. If it was 200 and 250, then that's not a big difference. But if it's two very different numbers reported by different sources, that is a large degree of uncertainty. And we recommend that you make two indicator calculations, in fact. And then you can use those upper and lower bounds as the boundaries of your uncertainty. And you can even do that all the way up to the country level, where you make one indicator calculation with all of your lower bound estimates, and then you make one calculation with all of your upper bound estimates, and then you have something in between. So I think that's the best way to incorporate uncertainty. It's not a feature of the red list. In the red list, I believe the precautionary principle is followed, where people tend to go with the lower estimate. So that is, I think, slightly different from the red list, and I think it's an advantage to include the full uncertainty. If the range includes 5,000, we do have a feature in the data collection device we created called the Kobo form, where you can choose that option. The range covers that, and basically there's a way to calculate so that some of those will be considered above and some below in the final calculation by kind of random choice. So I hope that helps and encourages you that even if there's some uncertainty, there's still a lot you can do with the indicators.
Okay, thanks, Sean. And then the last question in the indicator calculation session for Julia. It's about combining values across species. Suppose we have assessed the NE500 and population maintained indicator for a large number of species from different taxonomic groups. Do we get a single county value for the CBD reports by averaging across all species, or should we weigh them to account for each taxon, for instance, fish and birds, so that they contribute equally, or should we weigh by how common the taxon is in our country? So equal weights or weights according to commonness.
Yeah, so if you would just sort of take the average across all species, you would not really account for the bias you have, as the question sort of implied, right? Because you have different taxonomic groups, and then you also might have different numbers of species per taxonomic group. So I would say you want each taxonomic group to contribute equally and sort of rule out this bias of a taxonomic group. For example, mammals, if you have way more mammals than fish, for example. And there are a couple of options to weigh sort of these things. And what you could do is you can calculate the mean of each taxonomic group and then take the mean of these mean values for your country level indicator value. Sorry, that's a bit complicated, but I hope I expressed myself clearly enough. So that sort of downweighs this overly represented taxonomic groups. You can weigh species by the proportion of their geographic range within your country, for example. You can scale it or weigh it from 0 to 1. If you have endemic species, you could, for example, give it a weight of 1 to sort of really emphasize this species which only occurs in your country. And I think that's maybe preferably like give equal weights to each taxonomic group. And even though the CBD is requiring an overall country value, sure, it's also suggested to report a value based on taxonomic groups. And I think that's very wise. And I think to track change over time, we could even go a little bit further and say, let's report values per species, because that really gives you that high resolution of tracking the change over time and allocating resources and be a bit more active about it. Because you do have all these indicator values anyway, because you calculate it per species and then you average it. Yeah, I hope I answered the question now.
Okay, thanks, Julia. Sean, you want to add something?
Just to build on that, maybe we will share afterwards or in the chat. There are a couple of, we have at least one country that has already finished their reporting. So the UK recently published their indicator report online. So we could share that. The province of Ontario in Canada also did, and they did just as Julia said. They have it broken down by taxonomic group. And I think the one for the UK has also the full list of every species.
Yeah, it has the full list of all the species. Yeah, that's true. Thanks, Sean.
Okay, thank you. And with this, we reach the second part of the webinar. If you got any comments on how to handle cross-border populations?
Yes, if you have a species across 2 or maybe 3 countries, there are ways to join the information from these together, as long as you also have a political agreement with these countries to have a joint management. Because if you don't, and you just say, well, we have individuals from that country, we'll just add them to our own, and you don't have a concerted management or a population goal, it may be that the other one is just counting on you to reach 500 and vice versa. And the result is that both the population may decline in both regions because of that. So in short, I think this is also the case for the Habitats Directive. You can jointly report on a species for the Habitats Directive, but in some way you need to have an agreement across countries. And specifically with the Habitats Directive, there's been some jurisdiction on this recently with regards to that, where they say at least the countries need to be party to a similar convention, for example, the Berne Convention or the Habitats Directive. And if the countries are not, you can't join these populations together because they're subjected to completely different management strategies. So, for example, if you have, let's say, wolves in Russia and wolves in Finland, since Russia is not a signatory to the Berne Convention nor to the Habitats Directive, and it's a game species there that is treated as vermin, well, it doesn't make sense to join these together and count on Russia to do the right thing with the joint population.
Some species, like fish, may comprise many local genetically distinct populations connected by limited gene flow that likely cannot reach local NE larger than 500. How should such species be addressed in this context?
I'm always skeptical when somebody says cannot reach local NE larger than 500. I think there are many cases where they actually can reach, but it's a matter of wanting to reach. Yeah. With regards to fish, you can have local genetically distinct populations, but there are many very often nowadays migration barriers. They can get downstream but not upstream. And so specifically to that question, so the upstream subpopulation then is in fact isolated. And if it cannot reach NE500 because of this isolation, well, then that's just the case. The solution is solving the migration barrier downstream. And if you think further about this, suppose that the upstream subpopulation goes extinct because of it's too small, then the gene flow to the downstream population also decreases or goes to zero, and the effective size of the downstream population also declines. So in effect, you have this cascade of subpopulations that are interconnected, and where you risk to have a gradual failure or gradual loss of genetic diversity over time because of the cascading effects of upstream extinctions. Maybe if I hope that's clear. What we do see in terms of genetic diversity in such species is that you have currently accumulation of genetic diversity downstream because you have gene flow in one direction but not in the other. But the result is that the upstream populations are most vulnerable and are likely to suffer from inbreeding and inbreeding depression. Many of these migration barriers are relatively recent in evolutionary terms, 100 years, 200 years, maybe 500, but that's still recent. So if then the comment is it's impossible for them to reach NE500, well, then that is typically due to migration barriers and man-made migration barriers. I think it's important to assess the spatial scale of the population. So for some species like trout that live in short rivers but are connected to the sea, it could be that what your river is maybe just a subpopulation of a larger metapopulation. So that's something to take into account. We can't always know this prior to genetic data. There could well be very strong local adaptation to a very particular specific small river. And there are definitely cases where you have populations that might never reach NE500. But then probably these are on the long run kind of sink populations in evolutionary standards that depend on every few hundred years or so recolonization from a kind of mainland or main sea population.
Next question is, how can we deal with species that show large year-to-year changes in effective population size, such as beetles or grasshoppers?
This is a good one. This is where it may be good to have some collaborations with some genetic experts in calculating this. But there are known genetic methods to deal with fluctuating populations and to convert that to an effective size. Briefly, it's done by taking what's called the harmonic mean, which is an average, but it's weighted towards the lower numbers. And Joachim, you can correct me or add anything if you want, but I think that would be the typical way to do it, would be to take a harmonic mean of those fluctuations. What do you think, Joachim?
Yeah, I mean, at least for the census size, to have a kind of a correction there. Also, since when we use genetic methods, we are typically estimating the effective size of the parental generation. So that's something to keep in mind if you try to apply an NE to NC ratio. So if you have in 1 year a low number of individuals, but you sample them genetically, you're calculating the effective size of the parental generation, which could have been a much larger, or vice versa. So it's important to take this generational delay into account when doing this empirical calibration of census size and effective size. Now, with species like grasshoppers and beetles, I mean, if you can't get 5,000 individuals in such species, in such populations, then you really have an issue with biodiversity.
Then next question about, well, beyond the pilot studies that have been conducted in some countries, are there any examples of countries who have established an operational strategy and implemented concretely these indicators?
Yes, so that would be Belgium, also the UK, as you have seen, their report is already published even. Also South Africa. It will also be done in Mexico, and Switzerland as well. Those are the first ones that I can think of where I know the countries are already in the process of selecting species and making the calculations.
And I also think Finland sort of started selecting their species in a comprehensive way. So I think we're on a good way to it. It's just a process.
In Flanders, we're using these indicators to define for the habitat, to evaluate the situation of species for the Habitats Directive. So we're using indicators and proxies related to the NE 500 criteria to evaluate the conservation status of a number of species.
Question 8, some of my scientist colleagues are very critical about the indicators, saying that they're oversimplified. What should I tell them? And Sean knows the answer.
Thank you, Joost. I think this is a great thing, actually, to be honest. Biodiversity indicators have to run a middle line in between the rigor of science and the practicalities and simplicity that policy people need. And so actually to be criticized from the scientists for being too simple is probably a good thing. It means we are making something that is more on the line of being useful for practical purposes. This is actually the case. We have mentioned earlier today some comparisons to the red list. It's the case for the red list. The red list is an approximation of a species' likelihood of going extinct, and all the thresholds used in the red list are very simplistic and are not based on a super complex model built for each species. Typically, the biodiversity indicators and indicators largely, including for climate, are to be things that people can understand, that the public and policymakers can understand to some degree, things that can be done cheaply and quickly and at large scale, while still being meaningful. And we think to have some critiques from both the policy and science side means that the indicators are running that middle line, which indicators should and really must do. If you'd like to read more about that, we'll put a link in the chat to a paper, which is open access for all, and it was written for a non-scientist audience, so it's not too academic. It's called something like Too Simple or Too Complex. And is about this trying to answer critiques from both the science and the policy side. So I hope that's helpful.
Can you say something about the need for and how to keep the assumptions made during calculation linked to the data or the calculation itself or to the report? For example, in cases like the stork and partridge, which are not very straightforward. So how to keep a record of any assumptions made?
That's a great question. I can give a general answer and then maybe Julia can expand. So again, to compare to the red list, my institute, just like Joachim's and several others, is in charge of doing red listing. And when we do red listing, we compile like a document that has all of the information, the sources of data, who we talk to, what information we ruled out, extra information we didn't include, and assumptions and calculations, etc. So I think this would be done and is done in the countries we've worked with so far. So there's a document which has one or a few pages per species that has the maps, the data sources, and the assumptions. Julia, you are nodding your head. Would you like to expand?
I agree with you, and I just wanted to sort of tell about, because we're talking about the stork and the partridge, which is part of which are part of this bird project, and what we're also doing, as Sean now explained, we're also having sort of I call it a species fact sheet, where we do store all of that information. We also have comprehensive Excel spreadsheets in addition to that, to have sort of everything ready for the indicator calculation. And based on this project we're working on with the birds, we're trying to write like a workflow where we are describing sort of what information is beneficial to keep and how to have it in a broad like overview. And we hope that to share this workflow at some point soonish. And so then that maybe could also be a way for people to do it the same way, use it as a template.
Due to lack of resources, we can currently assess only about 20 species in our country. Should we still submit the report?
The answer is yes, for several reasons. The first is to set up the infrastructure and to just get the process started. So anytime you work on a new project or a new reporting mechanism, it's some work to get the data flows and the people in place, and just to practice. So the first reason is to get that started and to show that it can be done. And then you'll have a better idea of how to store the data and the time it will take and who you need to talk to for the next reporting. The other reason is it still provides some usefulness, both overall and for those 20 species. So it won't be as accurate as the recommended 100 species, but it's a start. And that number can still be useful for motivating action. And lastly, it's useful for those 20 species. So maybe those are 20 priority species in your country.
Yeah.
Julia?
Yeah, I just want to emphasize that only because you're sort of reporting this time on 20 species doesn't mean that the next reporting period you cannot expand, right? So that is sort of the goal, that if you now, for example, only have 20 species to report on, you then have 3 years time to set up the infrastructure for more species, or to really think about the species selection process. I was part of these workshops, the country workshops within GINAMO, and a lot of discussion was going also into sort of what species should be selected. And I think this is the first step. That's a very important step. And so probably a lot of countries are a little bit insecure about that right now. But again, I really think even 20, or I agree with Sean, even 20 species, that's a good start, because it really helps you to set up the infrastructure, get some responsibility sort of allocated to people who is doing what.
Is it necessary to calculate the indicators for all species within a taxonomic group to be unbiased? For instance, do we need all birds that occur in a country?
If you can, sure. But I mean, you're taking a sample. In statistics, you take a sample to say something about a whole. Here also, we can take a sample of species to say something about the total of the species.
I agree.
Yeah.
I agree. It's not necessary to do them all. If you can, great. I can tell you, South Africa has done all mammals in South Africa. It's something like 400, 450. So for some groups, it may be possible if you have the time and the resources and the data. But for many countries and most groups, it's not possible. But it can be done. The recommended minimum is about 100, as Joachim said, for a good statistically reliable sample. So that could be at the country level. If you really want to work with a particular taxonomic group, you may need that many for that. taxonomic group. But again, it's always a sample and typically will be much less than the full number of species.
And for many species, we use occurrence data based on citizen science for the assessment. Our current calculation of the NE 500 indicator is based on occurrence records collected over more than 10 years. Is this okay? Should the 2029 report only be based on records added between 2026 and 2029, even though that might be much less data?
I can start to answer. Joachim probably would be good to follow up because I think citizen science records are used a lot in their country. We always have a challenge of older data with biodiversity. Again, I've had a lot of experience, well, not a lot. I've had some experience with the red list process, and you often have to use older data, sometimes 10 or even 20 years since we last had an update on the knowledge from that population. And so that is what we use. Just to mention, we in the COBO data collection form that we created, you enter the date of when that information was accurate. So when the population was surveyed. So if you want to get advanced with your indicators, you can make an indicator calculation using all the data you have, and then maybe just using more recent data, for example. But typically in biodiversity indicators, we have to use all the data we have, including some quite old data. But Joachim, do you want to speak especially to the citizen science part?
Yeah, for the citizen science part, we use aggregated data across 10 years. So for our report, we used 2014 to 2024 approximately. So it's not necessarily limited to a particular year or a particular period. What you want to have is a good view of the distribution where they occur. And citizen science data are very prone to biases. For example, observer bias. There's more observers in some regions than in others. So this is something you want to take into account when you're using these kind of data. So it's an imperfect way to still get sensible information on the status of a population. That I think that's the core underlying thing we need to remember.
For many species, we roughly know the area occupied based on observation records. However, we do not have density data, and therefore we cannot calculate the census size from these observation records. So the question, what should we do to obtain effective population size estimates for the NE 500 indicator?
It's similar to the question earlier about clonal species. Earlier I talked about plants. Here, this can also apply to animals. We may have some observations that tell us about where a species occurs. Maybe it's some bird. And we know that a typical breeding size, a typical territory of a bird might be 1 square kilometer. Okay, and so you can take the total area from your citizen science observations where you know the species occurs, and then you can divide that area by the number of territories or a density estimate of typically, maybe you see 5 individuals per hectare. And so from that, you can get, again, an approximate of the census size and then the effective size. And just like I said before, there's some uncertainty in that, so you can perform several indicator calculations with different estimates. Maybe 1 breeding pair per square kilometer, or 2, or maybe 5 or 10 individuals per hectare or something like that. So yes, you can make some educated guesses about the number of individuals per area and then get to the indicator value.
So there are species for which this is going to work very well, and there are species for which it's not going to work at all. So some species are highly territorial and they have fixed home ranges, more or less fixed home ranges. So for these species, it will work pretty well. For other species which are gregarious, maybe, or live in large family groups which may vary in size, it may not work. So take this into account. But for some species, like, for example, amphibians, this seems to work quite well, although we do have correction factors per habitat type. For example, for the natterjack toad, we know that in coastal areas, population densities are higher than in inland heathland populations, which is 2 to 3 times higher. So we take that into account when we set then these threshold values. But most importantly, what we use a kind of average value in optimal habitat. So for example, in many amphibians, this is in the literature approximately 20 individuals per hectare. So you then know you need at least 250 hectares of this optimal habitat to have your NE500 population. So if your populations that you define based on where they occur in the citizen science data, if this is way smaller than 250 hectares, you already have your answer. You know then the population is likely to be much smaller than an effective size of 500. So this is how you can use these kind of approaches.
The experts we need to consult for the genetic indicator work are often the same people involved in red list assessment and other national conservation assessments. How do I convince them that it is valuable to spend their time on this?
I think this is great. And Joachim will follow up probably. But already we have several examples. For instance, like I mentioned in South Africa, they were able to calculate the indicators for all of those mammal species, 450, by collaborating with the National Red List process. So if you are able to connect with the National Red List process or other red listing processes, it takes very little extra time. So to have a form or a simple interview sheet, as the red list experts are gathering their knowledge for the red list assessment, you can very quickly, often, get the indicator calculation, the indicator values as well. So I think that's great. You can convince them by saying it should take very little extra time to discuss this, learn about it, and then get that data down, recorded. And then you have another really important indicator for the country. So it's a little bit of extra effort.
All right, thanks, Sean. Good. With this, we are going to end the webinar. So I want to thank again the panel, Julia, Joachim, and Sean, for all their useful insights. Every one of you in the audience, thanks for all your questions and also participating to the webinar. And then in the background, there's a lot of GINAMO members who help to prepare the webinar and sending invitations and preparing the questions. So thank you everyone for joining, and we hope to see you on the future occasion.
Yeah, and do reach out if you have questions. To reach out to the GINAMO email address, and they can dispatch it to us or other people and help you out with your questions and calculating indicators.